Hello everyone, my name is Anissa Nofita Sari. I am a third-year Ph.D. student in Tsukuba Life Science and Innovation, University of Tsukuba Japan. My supervisor is Professor Nuwatua from National Institute of Advanced Industrial Science and Technology Japan. On these occasions, I would like to deliver a presentation on my research. As shown in the title, I'm working on MortaparibPlus, a novel anti-cancer small molecule abrogating Mortalin 53 interaction in cancer cells. As we know, cancer is uncontrolled growth of abnormal cells in the body, and it is considered the second leading cause of death worldwide. There are several causes of cancer, such as heredity, UV radiation, and others. In cancer cells, these causes will trigger several response mechanisms, including the activation of tumor suppressor protein p53, which plays an essential role in preventing carcinogenesis by inducing cell cycle arrest or apoptosis. However, it has been reported that this protein might become inactive when interacting with the heat shock protein 70 called Mortalin. In cancer cells, Mortalin is highly expressed and bind to the p53 in the cytoplasm, preventing the translocation of p53 into the nucleus, hence the failure of tumor suppressor mechanism. Until now, there are several natural compounds known as inhibitor of p53 Mortalin complex, but in my study, I am focusing on synthetic compounds. To understand the potential activity of several synthetic compounds, we establish a screening step to search for potential abrogators of p53 Mortalin interaction. Using cancer cells with a wild-type p53, a chemical library was screened based on two criteria. First, enrichment of p53 in the nucleus, and second, change of Mortalin staining pattern from perinuclear to pancytoplasmic. After several cycles, we identified top 6 candidates. We had reported that the first novel anti-cancer molecules called Mortaparib and second novel anti-cancer molecules called MortaparibPlus. Then, the question is, why did we name them Mortaparib and MortaparibPlus? Mortaparib was named after its interesting dual anti-cancer mechanism. First, the inhibition of Mortalin that leads to disruption of Mortalin 53 interaction, and second, the inhibition of catalytic activity of DNA repair regulator PARP1. Mortaparib report cause apoptosis, cell cycle arrest, and metastasis in the cervical and ovarian cancer cells. Now, how about MortaparibPlus? Similarly, MortaparibPlus also has the mentioned dual inhibition properties. But what makes it plus? Interestingly, we found that not only did MortaparibPlus cause apoptosis and growth arrest in the p53 wild-type cells by downregulating Mortalin and activating p53, but also this compound activates apoptotic or growth arrest mechanism in the p53 mutant cell line by upregulating p73 and downregulating CARF. Let's talk more about how we discover MortaparibPlus. In this study, I start my analysis from the cytotoxicity assay in human colorectal cancer, breast cancer, and normal cells. I aim to study the potential activity of MortaparibPlus in different cell lines with different p53 status. I choose two cancer cell lines, colorectal and breast cancer, of similar cellular and molecular characteristics, with an only difference on the point mutation of p53. As shown in the figure A, HCT116, the LD1 and MCF7 cells were responsive to the MortaparibPlus, but only T47D that did not give any response on this molecule. From the morphology of the cells, in the figure B, we can see that T47D did not show morphological change compared to other cell lines. Then, to further examine the potential activity of MortaparibPlus on p53 Mortalin disruption, I conduct in-silico and in-vitro analysis. As shown in the simulation here, in figure A, MortaparibPlus could bind to the binding site of Mortalin and thus preventing the formation of P3 Mortalin complex. Through the in vitro analysis shown in figure B and C, P3 fractions in the Mortalin complex decrease significantly after MortaparibPlus treatment, regardless of the P3 status. The inhibition of Mortalin and P3 interaction by MortaparibPlus result in the decrease in Mortalin and increase in P3. This inhibition also enables the translocation of P3 from cytoplasm to the nucleus in HCT116, the LD1 and MCF7 cells, but not in the T47D cells. Now, we know that MortaparibPlus had a certain effect on Mortalin and reactivate P3. So, what happened with the carcinogenesis in the cells? Interestingly, I found that MortaparibPlus induced apoptosis. As you can see in figure A, MortaparibPlus caused an increase in the percentage of apoptotic cells only in the HCT116, the LD1 and MCF7 cell line. Molecular analysis also showed the pro-apoptotic proteins, PUMA and BEX increased in the three cell lines, but did not give any molecular change in T47D cells. Besides apoptosis, the reactivation of P3 protein also induced cell cycle arrest. I conduct cell cycle analysis to examine the potential activity of MortaparibPlus on this. As shown in the slide, MortaparibPlus caused G2M arrest and increase the P21 expression in the HCT116, the LD1 and MCF7, but not in the T47D. I have also confirmed that this molecule has the potential to inhibit PARP1 activity in HCT116, the LD1 and MCF7 cells, and resulting in the DNA damage. Here's a very interesting result. Remember from my previous explanations that T47D is resistant to MortaparibPlus? Well, despite being unresponsive and not showing either apoptosis or growth arrest, I found that MortaparibPlus works in unique mechanism in T47D cells. As shown in the figure D, E, and F, MortaparibPlus causes hyperactivation of PARP polymer, accumulation of DNA damage, and decrease in ATP level. These biological processes indicate that MortaparibPlus caused metabolic stress and exerted a different effect on T47D cell line. MortaparibPlus has multiple mechanisms in the cancer cells. As you can see on the screen, there are three mechanisms regarding to how this compound works. First, in the HCT116 and MCF7 cells, MortaparibPlus causes apoptosis and growth arrest by activating P53, as well as downregulating Mortalin and PARP1. Second, in the DLD cell line, MortaparibPlus activates P53 independent mechanism through the upregulation of P73 and downregulating of CARF. And third, MortaparibPlus induces metabolic stress in the T47D cell line. MortaparibPlus inhibits the interaction of Mortalin with P53, cause apoptosis and growth arrest, and induce metabolic stress. MortaparibPlus is a novel multimodal candidate anticancer drug that warrants further experimental and clinical attention. Finally, I would like to express my great appreciation to my supervisor, Professor Anuwatwa and Professor Sunil Cole for the valuable suggestions during the planning and development of this research. My special thanks also to my team members for the wonderful teamwork, Ahmad Elwakil and Dr. Jasprit Kaur, and all AST India Dialog members. Thanks to UTGRV School of Medicine for giving me the opportunity to present my research result at this international conference. Thank you for your attention.